Alright, so picking up with the renal corpuscle. If you look, there's a lot of cool stuff in here, like these juxtaglomerular cells and this macula densa that you'll learn more about in physiology and how they control the urine output and concentration of the urine. What we're going to talk about is as your blood flows into this capsule, you'll see it's two layers. You have this first layer of these cells that make up the capillary. These cells are a special type of simple squamous cells, and so it's just one layer of those really thin, odd-shaped cells. And these are called podocytes. If you look, they kind of look like they have pseudopods, where they interlock. When they interlock, there's actually a lot of gaps in there. So it's a fenestrated capillary. It's a capillary with a lot of holes. And it allows for this capillary to be under a little more pressure and to push all of this stuff into this area that is also, if you look, another layer of simple squamous, makes the outer part of the capsule. And in here is where you're going to get all that superfiltrate. And then it goes down into the tubules. So the first one is your proximal convoluted tubule. You'll see it is made up of simple cuboidal epithelium. And the simple cuboidal has microvilli. Alright, so here's the glomerulus, here is that capsule, and then we get to our first one, your proximal convoluted tubule, simple cuboidal with microvilli. Why does it have microvilli? To increase surface area. And what is happening is right here, after you get this superfiltrate, it comes in here and we start reabsorbing stuff. And so we pump out like everything we can, and then we start pulling things back in that we need. And so that starts happening here at the proximal convoluted tubule. Then we go down to the loop of Henle, or the nephron loop. And you're going to see you have this thick segment and a thin segment. The thick segment is where you're going to get a lot of salt transport, and so you're pumping out a bunch of salts that you want to keep in your body, so you don't want to get rid of. So you're actively transporting salts back into this area. And then this thin segment is really permeable to water. So when you're pulling salts out, water is a clingy friend. It wants to hang out with the salt. And so as the salt gets more concentrated out here, the water also flows out. And so that helps to concentrate your urine and help you to retain more water. Then we get down to the distal convoluted tubule. Also, simple cuboidal epithelium, but this time it doesn't have microvilli. And so if you're looking at a kidney slide, if it looks like the cuboidal cells have little brush border or hairs on it, those are going to be proximal, where the ones that don't are going to be distal. And then the distal goes into a collecting duct. Those collecting ducts go to the tip of the medullary pyramid at the papillae and empty into those minor calyces. Alright, so in order, which one's first? Glomerulus, major calyx, arcuate artery. It's going to go the artery to the glomerulus and then to the calyx. Make sure you know your blood flow through the kidney. I can promise you I'm going to ask at least a couple questions over this, okay? So, renal artery to segmental to interlobar, right? Arcuate, interlobular, no. And then trace the route of urea as it's filtered from the bloodstream. So if it is in your afferent arteriole, it goes to the glomerular capillaries, then it gets filtered out into the capsule, proximal convoluted tubule, loop of Henle, distal convoluted tubule, to your collecting duct. Those collecting ducts empty into the calyx, to the minor calyx, to the major calyx, to the renal pelvis, and then the renal pelvis empties into your ureters. So you're going to have a pair of these. You're going to have one on the left, one on the right. They are also retroperitoneal, so they're outside of that body cavity. And you're going to see 3 layers to them. So you're going to have an adventitia, which is similar to the serosa that we saw in the gastrointestinal tract. Then you have a layer of smooth muscle called the muscularis, and then finally the mucosa, and this epithelium is going to be transitional, so it can stretch. The muscularis does do peristaltic contractions to help force the urine down into the bladder, but it doesn't have to do it really hard because most of it is just going to be done by gravity. But occasionally you can get kidney stones, and those stones can be blocking the ureter, and then you're going to have those peristaltic contractions where it's forcing that stone down into the bladder so you can get rid of it. We also see a little flap-like valve right here at the end of the ureter when it empties into the bladder. So this little valve, you're just going to be trickling urine down into here, but then when you go to squeeze your bladder, that forces this valve shut. So it's pretty similar to the semilunar valves that we see in the heart, where as it pumps up, it opens, and then as it comes back down, it closes, right? And so this behaves pretty similar to that. So when you squeeze it, it's going to shut this valve. And so the urine goes out the urethra and not back up into the ureters, because that would cause a lot of pain. So when we look at the bladder, you're going to see it's really just this big sack of muscle, all right? It has 3 layers. You have your detrusor muscles. These are going to be important in the stretch reflex. So as your kidney, or as your bladder stretches out, those muscles have stretch receptors that send a signal to the brain that says, hey, we're full of urine and we need to be voided. That normally happens around 500 milliliters. You'll also see rugae in the bladder lining it. Same reason that you see rugae in the stomach, right? So it helps to allow it to stretch even more. The epithelium in the bladder is going to be transitional as well, so it can even stretch more. So you get lots of stretch here in the bladder. Here you can see the ureters coming in from behind posteriorly and then comes up inferiorly. Right here to open up. And we get this thing called a trigon between the 2 ureter openings and the single urethral opening. And in this trigon, you're not going to see any rugae, which is just kind of neat. Then you come down. The bladder has 2 sphincters, an internal and an external. Internal sphincter is going to be smooth muscle. External sphincter is skeletal muscle, so this is the one that is controlling everything. And then you have your urethra. Looking at the urethra, females, they have a really short one, 3 to 4 centimeters. This can lead to a lot of UTIs or urinary tract infections because it is very short compared to the males. If you look at a male urethra, it's about 18 centimeters long, give or take, with 3 major regions. So here you have your urinary bladder, and then all of that is urethra. Here at the prostate, you're going to have your prostatic urethra. And then as you come down, you have your membranous urethra, about the same area where you have bulbourethral glands. And then lastly, the longest part, the spongy urethral, and this is going to be found in that corpus spongiosum of the penis, which we will talk about more when we get to the reproductive system later this week. Alright, so where are the detrusors located? They're located on the bladder, in the bladder, and their job is to, one, squeeze the bladder when it needs to be squeezed, and two, to send that stretch signal to the brain that tells you that your bladder is full. Compare and contrast the internal and external bladder sphincters. Internal is made of smooth muscle, external is skeletal muscle. And with that, we are done with the urinary system.